Hello viewers, in this video we have discussed about complement system. The complement system consisting of over 30 proteins produced by the liver and found circulating in the blood serum and within tissues. Complement system is a part of the immune system that enhances the ability of antibodies and phagocytic cells to clear microbe and damage cells from the organism and promotes inflammation. It is a part of innate immune system. The complement system was first discovered by Jules Bordet in 1895. He established that the immune bacteriolysis and hemolysis requires two factors. One is eat stable antibodies and eat labile components alexin. The immunoglobulin IgG help to the immune system to trigger the complement system. The term alexin was renamed as complement, which was coined by Paul Ehrlich. Complement is eat labile in nature. The complement does not bind with free antigen or free antibody. It can bind only with antigen-antibody complex. The complement system consisting of over 30 proteins produced by a liver and found circulating in the blood serum and within tissues. Functions of complement system. The first function is defending against bacterial infection by enhancing phagocytosis through oxonization, hemotaxis, inflammation, activating leukocytes, and lysis of bacterial cell wall. It results cell death. The second function is bridging innate immunity and adaptive immunity by augmentation of antibody response and enhancement of immunological memory. The third function is disposing and clearance of dead host cells. Components of complement system. Recognizing units is C1 and activation units are C2, C3, C4, and then membrane attack unit is C5, C6, C7, C8 and C9. Alternative pathway having two factors that are factor B and factor D. Regulatory proteins are C4 binding protein, C1 INH complex and factor H, factor I. The classical complement pathway can be initiated by the binding of antigen-antibody complexes to the C1q protein. The globular region of C1q retinizes and bind to the surface of Fc region antibody isotope IgG or IgM. These globular regions of C1q can also bind to the bacterial and viral surface proteins, apoptotic cells and acute phase proteins. The following activation, a series of proteins are required to generate C3 convertase, which cleaves the C3 protein. The function of the membrane-bound C3 convertase is the cleavage of many molecules of C3 into C3a and C3b. C3a is a smaller fragment of C3. It is potent inflammatory mediator. And then formation of C5 convertase and MAC. C3b bind to the C3 convertase to form C5 convertase. C5 convertase then cleaves C5 into C5a and C5b. Like C3a, C5a is also anaphylatoxin that interact with its C5a receptor to attack leukocytes. Subsequent interaction between C5b and other terminal components C6, C7, C8 and C9 form the membrane attack complex or other C5b9 complex, which forms force on the target cell membrane to lysing. Complement system consisting three pathways, that are classical pathway, lectin pathway, alternative pathway. In classical pathway, antigen-antibody complex in the presence of C1, C4, C2 units to form C3 convertase. In lectin pathway, lectin binding mannose in the presence of MASP, C4 and C2 to form C3 convertase. In alternative pathway, pathogens injure tissues in the presence of factor B and factor D to form C3 convertase. This C3 convertase cleaves as C3a and C3b. C3b bind to the C3 convertase and form C5 convertase. Then C5 convertase cleave as C5A and C5B. C5B other terminal components C6, C7, C8 and C9 form the membrane attack complex or C5B9 complex, which forms force on target cells membranes to lysis. Thank you.